Dear students, Assalamu alaikum. Today I will deliver a lesson for class 12, subject zoology. My today's topic is polymerase chain reaction, abbreviated as PCR, and its applications. Now, polymerase chain reaction or PCR was initially developed by a scientist called Carey Mullis in 1985, for which Carey Mullis shared the Nobel Prize with Michael Smith in chemistry in 1993. PCR is basically the DNA replication in vitro. Now, during PCR, selective amplification of a specific region of DNA molecule takes place. Now, the basic requirements of a PCR are: number one, DNA template. Now, any source that contains one or more target DNA molecules to be amplified can be taken as DNA template. The second most important requirement is nucleotide primers. Now, primers are basically oligonucleotides, usually 10 to 18 base pairs long, that hybridize to the target DNA region, one to each strand of a double helix. Now, two primers are required, and these primers are oriented with their ends facing each other and allowing synthesis of the DNA towards one another. The third requirement of the PCR are the enzymes. Among enzymes, DNA polymerase, which is stable at high temperature, at a temperature more than 90 degrees Celsius, is required to carry out the synthesis of the new DNA. The polymerase that is used in PCR is called Taq polymerase, which is isolated from a bacterium called Thermus aquaticus. Now, the working mechanism of PCR. A single PCR amplification involves three basic steps, that is denaturation, annealing, and polymerization. Polymerization is also called as extension. Now, you can see this is shown in the sketch, the steps of denaturation. First, this DNA is heated up to more than 90 degrees Celsius, by which the two strands are separated. Then primers get attached to each strand, and they are oriented in such a way that they complete the formation of the DNA strands on both the strands of the DNA, from 3' to 5' or 5' to 3' end. Now, this is called the annealing. Now, after annealing, what is done? The DNA polymerase is treated along with the deoxynucleotides, by which the amplification of this segment of the gene takes place, and this is also called as the extension. On each cycle of amplification, this gets doubled, the DNA, target DNA, and after 30 cycles of amplification, we get almost about 1 billion segments of the target DNA. Now, we will discuss about the applications of the polymerase chain reaction. So, for the applications of polymerase chain reaction are concerned, some of the important applications of PCR are: number one, diagnosis of pathogens. Recently, PCR has found application in the diagnosis of various pathogens like HIV, that is the AIDS virus. It is because the PCR can detect the presence of the gene sequences of the infectious agents. Our second application is in the diagnosis of specific mutations. Polymerase chain reaction is also used to detect the presence of a specific mutation that is responsible for causing a particular genetic disease before the actual onset of the disease. Thus, by using PCR, we can diagnose the disease like phenylketonuria, muscular dystrophy, and sickle cell anemia like genetic disorders before the appearance of the symptoms of these genetic diseases. Our third application is in DNA fingerprinting. PCR has a great importance in DNA fingerprinting, which you have studied earlier. Fourthly, this PCR has a very important application in prenatal diagnosis. This PCR is very useful to detect the genetic diseases in a fetus before birth. Now, if the disease is not curable, then the abortion is recommended for such pregnancy on eugenic grounds. Another importance of the PCR is in paleontology. PCR is also used to clone the DNA fragments from the mummified remains of the humans and other extinct animals like dinosaurs. Lastly, in the diagnosis of plant pathogens, PCR is also used in detecting the various types of plant diseases caused by different types of plant viruses, bacteria, and mycoplasma, etc. Now, this was all about the polymerase chain reaction, its mechanism, and the applications. Thank you.